everybody, and welcome to Chapter 10. DNA, RNA, and proteins is what we'll be talking about in this chapter. And previously we looked at how cells divide and send their DNA to new cells and how the DNA inheritance patterns work. Well, now we're going to look at how, once we have our cells, how we get the DNA to code for the proteins. What we know about DNA is that it stores genetic info and passes it from generation to generation. Our nucleic acids, we touched on this in one of our early chapters. These polymers of DNA and RNA are made up of subunits called nucleotides, which are bound together by a sugar phosphate backbone and a nitrogenous base that codes for proteins. So here's the sugar phosphate backbone right here, this blue and yellow pole basically. And you can see it better in this picture with the 5-carbon sugar right here. In DNA, the sugar is deoxyribose, and RNA it's ribose. Then you have a phosphate group denoted with that little circle. And you have nitrogenous base right here. Remember, for DNA, the nitrogenous bases are A, T, C, and G. A binds with T, C binds with G, and it forms a double helix structure. Again, the 4 nucleotides in DNA that I just talked about. So this is touching on it again. These 4 are similar in RNA. But instead of thymine, we have uracil. So remember, in DNA, A binds with T and C with G. In RNA, anytime the DNA has a T in it, it codes for an A. And in RNA, anytime the DNA has a T, it codes for an A in RNA. And anytime the DNA has an A, it will code for a U in RNA instead of a T. There we go. Got it together. So now on to some history, some DNA history. Multiple studies try to figure out DNA's structure. They knew it passed down genetic information, but they didn't know how, and they didn't know what it looked like. Watson and Crick were the ones we credit with determining DNA as a double helix. They used X-ray crystallography, and Rosalind Franklin, this lady right here, was actually the person to collect the data. She was able to look at really, really small structures with this X-ray crystallography. And up until this point, other groups determined that DNA was some kind of helical shape, but they thought it had 3 backbones. Rosalind Franklin, actually found out that it was a double helix. Watson and Crick, and technically Franklin too, published this, and they won a Nobel Prize. Well, Watson and Crick won it. Rosalind Franklin died before she could accept one. She died of cancer at 37 years old due to all of this X-ray work, because at that time they didn't really know about radiation and its harmful effects and all that jazz. So they found that the model of DNA has a spiral double helix, so it's like taking a ladder and then twisting it. And there are many different artsy interpretations where you see the sugar phosphate backbone and the nitrogenous bases doing their complementary base pair thing. And remember that base pairs are held together by hydrogen bonds, and that these are pretty weak. So this is the best picture to see these in. Notice AT make 2 bonds right here, and then CG make 3 bonds. That's why it's important that the right bases are getting paired together. And now on to DNA replication. We know we have to send out a DNA template or blueprint or instructions to the next generation. So we're going to look at how that happens. And these are 2 different pictures of what happens during DNA replication, but I'll talk about that coming up. So the first step of any cell division is to copy the DNA. Remember, we doubled the DNA during S phase and interphase. And here what happens is the backbone, what happens is the backbone of the DNA gets split open. DNA helps open it up, or DNA helicase helps open it up. Helicase unzips the genes, if you will. And then we start matching new strands to each of the original strand. The original strands are blue, the new strands are this orange color. And this is all called semi-conservative replication because we're saving one of the old strands on each side. We're not going to be starting all over. We're still keeping some of that old parent DNA molecule and just attaching new daughter strands to make new strands of DNA. There are specific sites where you start DNA replication. These are aptly named the origin of replication areas, and it goes in both directions, as you can see here. And by the end, you got 2 new strands of DNA. They're exactly identical as long as there are no mutations, and there are lots of checks with DNA replication to make sure we have no DNA mutations. More on mutations at the end of this chapter. Alrighty, so we've mentioned DNA to RNA to proteins before in one of the early chapters, and now it's time to get more in depth with this. DNA is the genetic code that gets passed on, and we send it out into the cell through RNA, and at the ribosomes we make proteins. We bind amino acids together, so we go from this nucleic acid language to amino acids and make proteins. Remember, the genotype is the code and the phenotype is what it looks like. Here's just a good example of some different traits. So moving on, there are 2 stages. These 2 stages are called transcription and translation. DNA transcribes to RNA first. Transcription means to rewrite in the same language. And here it's going from this nucleic acid language in DNA to this nucleic acid language in RNA. Then secondly, we have RNA translating into proteins. Proteins are not nucleic acids. So they must translate to a whole new language of amino acids and then eventually proteins. In this process, this is called the central dogma of biology. So if you hear the phrase central dogma, this is what we're talking about. The one gene hypothesis states that for every gene it codes for a single protein. So you used to think it was a one gene, one enzyme hypothesis, but there are a lot of proteins that are not enzymes, so we fixed that. And we're now going to go through transcription and translation. First, what is the language of nucleic acids? It's these nitrogenous bases, the nucleotide bases, the AA, CC, GG, CAAA. That is the code right here. And from that DNA strand, this code right here, we make matching RNA bases based on the coding again. Remember, A binds with T, but in RNA it'll be U. So AAA would be UUU, the CC would be GG, GG would be CC, C would be G, and then AAAA would be UUUU, because this one's going to be the RNA. So here we have DNA strand blue, the RNA strand is red. Then based on this set of nitrogenous bases, they will be coding for an amino acid. And this is an example of polypeptides. So remember, lots of peptide bonds. Polypeptide, it's an amino acid chain. This is an example of an amino acid chain, and we'll talk about codons here shortly. But DNA transcribes to RNA, and RNA translates to proteins or to amino acids in a polypeptide. And remember that a polypeptide, again, is a small protein. So a little refresher about the bonds between amino acids being peptide bonds. So I was back to one of our earlier chapters on proteins. So if you need a refresher, go back and read through that chapter. So when the DNA is transcribed, the result is an RNA molecule. Again, here's that central dogma I was talking about, DNA to RNA to protein. And what is the correspondence between the nucleotides of an RNA molecule and the amino acids of a polypeptide? So we've seen there are 20 different amino acids, going back to our protein chapter, but we only have 4 different nitrogenous bases. How do we get this done with only 4 letters? Well, we need to have triplets of nucleotides on nitrogenous bases. So a set of 3 in a row codes for a single amino acid, and those are called codons. And that means that we have enough codons to code for all 20 amino acids. This genetic code, that codon, is shared by all organisms, so we are all technically related. In transcription, we lead those nitrogenous bases or nucleotide bases, and we make an RNA template. We don't move DNA over and have it become RNA. We open up the DNA, we make matching RNA, because remember, they're different structures. They've got different sugar, deoxyribose in DNA versus ribose in RNA, and they've got different bases with the U versus the T. And this here is a nice little view of what's happening. We split open the DNA. We have RNA polymerase that helps us. In DNA, it was called DNA polymerase. And this RNA polymerase helps make the RNA. It adds the RNA nucleotides in. So now the RNA polymerase binds on, and we have this initiation, elongation, and termination to make a completed RNA sequence. First is initiation. That's the start. The RNA polymerase attaches to a promoter, and this is exactly what it sounds like. It promotes RNA polymerase to come in at the beginning of a gene, and that's how we start RNA synthesis. That's how we start making RNA. Secondly, we have elongation. So we keep reading the DNA. We're adding more and more RNA nucleotides until we get to termination. So once you find this terminating sequence, the RNA polymerase detaches and it's done. So now we process the RNA. This is what we have to do to RNA before it can go to the ribosomes. DNA has different codes like exons and introns, and we have to cut out introns. Introns are bad. We want to get rid of them because the exons are the ones that become the protein. So we take out the introns and we glue together the exons. And then so introns removed, exons spliced together, glued together. And we add a tail at the end, and we send it out of the nucleus and into the cytoplasm towards the ribosome. So now translation. We have RNA, and now to make a protein. First, mRNA is needed. The M stands for messenger, and this carries the message for the code of proteins. And there are 3 different types of RNA, but so far we've only focused on mRNA. Another type is tRNA, transfer RNA. This does the actual translating. It comes in, takes the RNA code, and matches it with the anticodon. It takes the RNA codon and matches it with the anticodon. Right here on the end is where you can see anticodon, anticodon. And it's got an amino acid, so it comes into the mRNA, matches up in the right spot, and leaves the amino acid. And then we have rRNA, or ribosomal RNA. Ribosomes are made up of rRNA and proteins, and this right here is the actual site where making proteins or protein synthesis occurs. This ribosome. This is a picture of mRNA going through the ribosome and tRNA coming in when it's turn to add on some amino acids. Translation also has those three phases, that initiation, elongation, and termination stage. And so in initiation, we have the first amino acid bind, and it's always methionine. MET is how you'll see it. So here we have a cap and tail that help it bind to the ribosome. And then translation happens and proteins are made at the ribosome. The blue part right here is part of the ribosome. To start, you have MET, methionine. The first amino acids bind to the mRNA and come into the small unit of the ribosome. Next, or small subunit, sorry. Next, the large subunit comes in, and we've got the setup ready to go. The tRNA comes in through the A site and shifts over to the P site. And that's what we look at during elongation. So codon recognition happens, matches the codon with the anticodon, and the first codon, the start codon, will always be AUG. And the start codon, AUG, always codes for the methionine, the MET that we saw during initiation. This keeps happening to make peptide bonds, and we keep moving down to make more amino acids. So we're elongating it, we're making it longer. This is a little diagram, a little visual of what we just talked about. So codon recognition, peptide bond formation, translocation, mRNA movement, and then you see this little chain of amino acids being spit out. And last we have termination. So we do all of this until we reach a stop codon, and there are a few different codons that code for stop, but once we reach that, it's over. We are done. Termination. After that, we've got a primary structure of a protein, and next it can become secondary, tertiary, or quaternary. So if you want more information on that, you can reference our chapter over proteins. And this is a good overall review picture. Notice how DNA can go to replicate, or it can go on to transcribe, to transcription. And here's another review picture. Over here we've opened up our DNA. We've got RNA polymerase coming through. It's reading that DNA. It's matching it with corresponding RNA. Then we process that RNA. We cut out the introns. So introns are being cut out here. We add a cap and tail, and then we leave the nucleus. We go to the ribosome. We have the initiation once MET, that methionine, combines on an mRNA at a ribosome, and then elongation, where we keep binding tRNA to mRNA, adding on amino acids, and shifting over until there's a finished product. So we find the stop codon, and we have termination. In eukaryotic cells, transcription is going to happen in the nucleus, and translation will be in the cytoplasm where the ribosomes are. So the ribosomes are going to be in the cytoplasm, and then it's where translation happens, whereas transcription is inside of the nucleus. In prokaryotes, there's no differentiation. It's all going to be happening in the same space. So think about different cell structures. So transcription and translation, again, are the processes where we take DNA and RNA, make proteins, and those proteins are super important. They can be for cell structure, cell enzymes, and they determine our cell function. Now let's look at mutations. This is any sort of change in the nucleotide sequence, any changes in the nitrogenous base pairs. The example here is our recurring example of sickle cell anemia. Just one change of amino acids changes the shape of hemoglobin. There are two different types of mutations. The one in sickle cell is a base substitution mutation, where a single nitrogenous base is changed. Sometimes these can be silent, where they don't change the amino acid sequence, but sometimes they do. Obviously here you can see that this one base being different changes the amino acid, the protein. And that's what happens in sickle cell anemia. And then we have insertions and deletions, and these are really bad mutations. If you take one base out, for example, the whole frame shifts. Same thing if you add one in, the whole frame will shift. And all the amino acids following that nitrogenous base deletion or insertion have changed. So notice the original right here. We take out this U, and everything has shifted over, and the sequence has changed, and the codons are made different amino acids, have coded for different amino acids. And generally, whenever this happens, that will cause the protein to be non-functional. Every time we go through DNA replication, we have errors, but our DNA polymerase catches them. Well, it catches almost all of them. It only misses maybe 1 in 100 million. Viruses don't have polymerase checking it, so they have a rate of more like 1 in 100,000 base pairs being wrong. So we do have these checks, but we also have chemical and physical agents called mutagens that increase the rate of mutation. So you can just read through some of these examples right here. This is an example of some kids that were born with mutations that were causing their moms from anti-nausea medication they were taking back then while they were pregnant. Pretty scary. So mutations we've looked at have generally been found to be harmful. However, they are so important and responsible for genetic diversity. Where most mutations are harmful, you'll have one once in a blue moon that won't be, that will actually be selected for. And that'll give that organism an advantage, and that organism will pass it on to its future generations and hopefully give them advantages as well. And this is what makes the world super interesting and diverse, which is awesome. We don't all want to be the same. So last topic is viruses. Viruses are really interesting. We actually don't completely know if viruses are alive or not. There's arguments for both. Most people just sit on the fence and say, I don't know. They can do everything that we talked about in chapter 1, but they can't do anything without a host. So they need a living host to be able to function, to do something. Viruses have genetic material and packaging, but that's about it. They infect host cells and they make them into virus factories. So they have either DNA or RNA, and they insert it into a host cell, and that host will start making those viral proteins. Some RNA viruses are called retroviruses, and these are some of the more popular ones you've heard of. I'll talk about that in a minute. But some are called retroviruses. They take the RNA and they make it back into DNA before going through DNA RNA protein. And that backward step makes sure that there are no checks to ensure we're not making mutations. So a really popular retrovirus, HIV, that we've all heard of, it mutates at a much higher rate than regular viruses, and then the infected cells generally produce viruses until the virus exits the cell and the cell is killed. In our bodies, we have some pretty cool responses to viruses. For example, we have these T cells, which are some of our white blood cells that do immune function. And when these T cells get infected with a virus, they start making viral proteins. However, they also make a protein called interferon, and these interferons are going to go out and warn other cells like, hey, it's too late for my cell, but protect yourselves. And they put up a block so that the virus can't get into those other cells. Viruses are just super interesting, so we had to touch on that while we were talking about everything else. But that is all for this chapter. See you next chapter.